Hi, my name is Payton Aloi, and my project is on the analysis of mouthwash. The two mouthwashes that I analyzed were Listerine Coolment Antiseptic Mouthwash and Crest Scope Outlast Mouthwash. My project was a three-part project, and my first part is spectroscopy. So what is spectroscopy? In very simple terms, it is light interacting with matter as an analytical tool. In my experiment, I used a spectrometer to identify the wavelength and absorbance of dyes. Some definitions to know are absorbance, transmittance, and wavelength. Absorbance is the amount of light that does not pass through the sample. Transmittance is the amount of light that does pass through the sample. And wavelength is the length of two successive peaks. This is an electromagnetic spectrum, and the section of the spectrum that we'll be focusing on is the visible region. The image shown is an example of an absorbance spectrograph. Absorbance is on the y-axis and wavelength is on the x-axis. So how do you read and interpret an absorbance spectrograph? Well, after observing the graph, the line in which the color and the absorbance is the highest, opposite of that on the color wheel, is the transmittance. The absorbance spectrums that I analyzed were from dyes green 3, red 3, yellow 6, red 40, yellow 5, blue 1, and blue 2. On the following slides are the spectrographs of each dye. So now it's time to incorporate our first mouthwash, which is the Crest Scope Outlast. So the dyes on the label that were listed in the ingredients are blue 1 and yellow 5. After analyzing the Crest Scope absorbance spectra, I was disappointed to notice that it did not have a high peak like those of blue 1 and yellow 5. So what I did was I decided to concentrate the mouthwash. These images show the unconcentrated Crest Scope mouthwash spectrum compared to the Crest Scope concentrated spectrum. So once we compared the concentrated spectrum to the blue 1 and yellow 5, we notice that the graph does incorporate both blue 1 and yellow 5, staying true to the label. Moving on to the Listerine mouthwash, the label had listed that it only had dye green 3. After comparing the Listerine and green 3 graphs, it is evident that they are very similar, except the absorbance on the Listerine is not as pronounced. Moving on to part 2, this next part is all on column chromatography. So what does column chromatography do? Well, it separates mixtures and detects trace components of a mixture. A spec pack cartridge, which I used, is filled with solid nonpolar material, which is also the stationary phase. This table shows the steps, the solution I used in each syringe, and the color expelled in the solution. So my conclusions are, as the isopropanol alcohol percentage increases, the solvent becomes more nonpolar. Blue 1 is more polar than yellow 5 since it is in the column longer than yellow 5, while the increasing nonpolar solvents are added. Lastly, my third part was determining ethanol concentration in the mouthwashes. The equipment I needed for this procedure was a 250 milliliter flask, a burette, a 5 milliliter small glass vial, my 2 mouthwash samples, pipettes, and an incubator. The solutions I needed for this experiment were acid dichromate solution, starch indicator solution, sodium trisulfate solution, and potassium iodide solution. First, I had to prep the samples. For the Listerine, I diluted the samples to a 1:80 ratio, which is 12.5 milliliters and 1,000 milliliters of distilled water. For the scope, I had to distill the samples to a 1:50 ratio, which is 20 milliliters and 1,000 milliliters. My first step was to transfer 10 milliliters of acid dichromate into a conical flask, pipette 1 milliliter of distilled mouthwash into a glass vial, suspend the sample over dichromate solution, and add a rubber stopper on top. Now for this, I repeated this 3 times for both mouthwashes, which is a total of 6 flasks. Step 4 was I had to store the flasks in an incubator overnight at 25 to 30 degrees Celsius. Step 5, rinse walls of each flask with distilled water and add 100 milliliters of distilled water and 1 milliliter of potassium iodide solution. Then mix that. My 6th step was to prepare a total of 6 blanks, which was 10 milliliters of acid dichromate solution, roughly 100 milliliters of distilled water, and 1 milliliter of potassium iodide solution. And then I mixed that. Step 7, fill a burette with sodium trisulfate solution and titrate each flask. Tighten treat until the brown iodine color fades into a pale yellow. Step 9, then add 1 milliliter of starch indicator. Step 10, continue to titrate until the blue color disappears. This table shows the data collected from the blanks. This table shows the data and results from the Listerine samples. And lastly, this table shows the results from the scope samples. So, is titration a good way to determine the percent of alcohol in a solution? Yes, but errors during the lab could curve the results. The label on the Listerine mouthwash said 21% alcohol. After doing calculations, the percent I calculated after titration was 24.21%. The percent of alcohol on the scope label was 13%. After my calculations, I calculated that after the titration, the percent was 15.63%.